Yes, welcome to the day 11 session on applications, that is on bioinformatics applications in systems biology and synthetic biology. So today we discuss more on system biology and what do you mean by system biology markup language, and how about network biology, network pharmacology, and we discuss types of networks. We discuss on synthetic biology, and then synthetic biology open language. So we discuss all this, including the softwares used for system biology and synthetic biology. Before proceeding on today's session on applications of system biology and synthetic biology, can we recap the last session contents? So as usual, we can discuss the last session topics. Very good. So I got an answer. So I think the net problem. Yeah, actually, sorry, there is some internet connection problems. Uh, Now I just, am I audible to you, everyone? Yes, sorry, actually, something has happened. The participant has answered the Gaurinayak that about the last session topics. What exactly did we discuss? We discussed yesterday on, very good. Uh, so on from a participant we discussed on protein structure validation. We have discussed on rmst called as root mean square deviation, and how to assess the stereochemical quality of a protein. The stereochemical quality of a protein is related to the secondary structure. If you want to identify the secondary structure elements, what we do is we use phi versus psi plot. Like whatever we find in the peptide, so we take phi versus psi and we try to get a kind of identify the secondary structure elements through Ramachandran plot. And so we assess the stereochemical quality. Later we discussed on the gene prediction softwares, and we have discussed on the two types of softwares are there for gene prediction. One is for prokaryotes as well as for eukaryotes, and we have seen plenty of examples for gene prediction. Okay, so this is what we have discussed on last session. Now let me take you to this today's session on system biology. So if you see in system biology, this is actually we use generally for bringing a precision based medicine. Like if you see for the diseases like cancer, we don't have precise medicine. Like whatever drugs available in the dispensaries, that is available in the market, can able to cure 1 in 3 to 1 in 20 patients based on the types of cancer. So if say now itself I have joined, it's very late to join. Participants don't join at this time. Okay, if you want to join, join earlier. Okay, now let me go ahead with the system biology. I was explaining you that if we are discussing on the system biology is majorly used for developing the precision-based medicine, where here we consider the genes, not just a single protein target, and of course not a single gene. We focus on one hub gene connecting with at least 10 interacting genes. Similarly, even for the proteins. Okay, so like however I mentioned earlier, so targeting 1 in 3 to 1 in 20 patients is based on types of cancer. Okay, it's very hard to treat the patients. So to reduce this kind of a complications, instead of focusing on a single target, we focus on network of proteins which we find in the networks. Okay, so here if you see in system biology, this is a combination of different omic studies, genomics, proteomics, metabolomics, transcriptomics, gene networks, okay, and molecular structure. And of course, we integrate the types of pathways to understand the disease information. Now, let me come across the system biology approach that we generally use the omics tools. The major system biology approach, okay, in earlier up to 2005, you know that most of the important genomes are published. And later, you can see from 2005 to 2015, many proteomes are published. So up to 2015, you can find at least some proteome databases are available. And later, the experiments were carried out in the metabolomics. You know, up to 2020, we are getting so many kind of a metabolites deposited in the metabolite databases. And then now in transcriptomics, still this era, we can see so much kind of a transcriptomes. Okay, that is RNA or getting deposited in this transcriptome database. Okay, so all these omics are integrated. Nowadays, just restricting up to the genomics, we can't able to bring up with the precision-based medicine. We have to link even proteomics. Okay, whatever we see in the past 2 decades, you can see so much kind of developments are happened, and even metabolomics, transcriptomics. Nowadays, we have to use everything. Okay, for finding the precision-based medicine. And if you see this relationship between the single and multi-omics analysis, okay, other than the genome, transcriptome, proteome, metabolome, now we can also see something called epigenome. Okay, in genome, these are some of the technical terminologies we can see: single nucleotide polymorphism, rare variant, genomic rearrangement. But whereas here in epigenome, we can find miRNA, histone modification. So, where you can observe the PTM, post-translational modifications. In that, you can able to see, especially whatever you study in genome with respect to PTM. That's what we call it as an epigenome. Okay, like this, there are some technical terms like pan genome. So all these things we discuss more about the types of genomes we discuss even on the 14th session. Okay, so this is just I'm mentioning you that in system biology we integrate all the types of omics. Okay, the DNA, RNA, protein, everything is important, including the metabolites produced in the pathways. Now about SBML. What is SBML? SBML stands for System Biology Markup Language. Like how we have HTML, XML. This language actually used to develop the system biology components. This is initially released on 2001, and file name extensions are .xml and .spml. So system biology markup language can also be written in database language. What is XML? XML is extensible markup language, isn't it? So this is actually a database language. So if you want to include a data inside the user defined tags of XML, we can use this, we can integrate the SPML with the XML. Okay, again I'm telling you the system biology markup language is a representation form based on XML for communicating and storing computational models of biological process. It is a free and open standard with widespread software support and community of users and developers. So generally the system biology markup language is used to represent many classes of biological phenomena, including metabolic networks, cell signaling pathways, regulatory networks, infectious diseases, and others. So this is something about system biology markup language. And if you see about the system biology markup language, this can be the information we can get from this spml.org. And for the softwares, we have to go to this URL. Okay, and if you see this system biology markup language, these are some of the softwares like libsbml. This is a library for reading, writing, and manipulating files and data streams in HTML format. It is written in C++ as well as in C and provide APIs for almost all the languages, whatever we use in bioinformatics. C++, C Sharp, Java, JavaScript, MATLAB, Octave, Perl, PHP. The MATLAB is popular software where we use the SPML to develop the system biology components. Okay, so in this session we also discuss something about the MATLAB. The other things, whatever mentioned here, are the languages. Even in MATLAB, we have the languages, but we also have components built inside the MATLAB so that we can able to build the system biology models as soon as possible, and it is easier for us when compared to following the programming languages to develop. Okay, and similarly, if you see this is a JSPML, this is another kind of tool. Actually, this is a pure Java library for reading, writing, and manipulating SPML. Okay, and the third is the divisor, and fourth is SPML test suite. So like this, we have plenty of softwares in system biology markup language. For this, we have to go to the URL mentioned below. What is system biology and how can it can be integrated? If you see, system biology is like a ocean. Plenty of information is there in system biology. Majorly, the system biology can also be used to study about DNA computing or cellular computing. So whatever we call cellular computing, instead of cell, if you study about DNA, we call it as DNA computing. And if we study any of these biomolecules, we call this as biomolecular computing. Okay, the molecules, it can be a molecule or it can also be, so in cellular computing, the cellular computing is something, a overall thing, where in the cellular computing, if we study about the biomolecule, we call it as biomolecular computing. And we can also study in the biomolecule gene and protein. Other than the biomolecules, we can also study organelle, that is a cellular subsystem. For example, mitochondria. It can be chloroplast. Generally, in human, you study about the mitochondria for cancer diseases. If you are working on cancer diseases, we study about that. And if you are working on plant kind of a metabolism, of course, we need chloroplast. So these are some of the organelles. Okay, we have so plenty of organelles in the cell, and whatever cell we take, I mean, whatever organelle we take, we can also study the computing approach, the cellular computing approach. We can also study the cell itself. That's what we defaultly call it as cellular computing. Other than the cell, we can also take an organ, for example, liver, and also an whole organism we can take, for example, human, and like this we can study. Okay, see, if in this example we have taken a cell, okay, a bacterial cell. In the bacterial cell, we just like a CPU, we can also call this as a neuron. In neural network, this can be considered as a neuron, a processing unit, where we feed inputs and we can derive some outputs. For example, here in a bacterial cell, if you feed lactose, so the lactose consuming bacteria can able to produce fluorescence based on how much lactose is utilized. Similarly, the light, based on light also, you can able to measure the fluorescence, how much light is taken by the bacterial cell. Similarly, we can see tetracycline quorum sensing. Whatever inputs we give, we can get the output of the quorum sensing. And similarly, based on the light, even the pigmentation of the algae or pigmentation of the plant. So everything it varies. Okay, so not just whatever we find in the plant, even the bacteria can also can produce pigmentation just like a plant. So based on the light, even the bacteria can produce pigmentation, especially if you see Pseudomonas fluorescens that can produce the fluorescence. Okay, and also you can find the pigmentation. Okay, and we can able to see even the bacterial cells can also produce the electric current, but that is based on the temperature, pH, etc. So what you can understand is in the computing, in a cellular computing, it can also be a biomolecular computing. Okay, we feed some inputs and we give a kind of a correlated formula, and whatever correlation formula we calculate in this neuron or a cell or organ, whatever we keep inside, okay, we'll be getting in corresponding outputs, and how much output we can also find in the computational itself. This is like a simulation. It's a simulation thing. Whatever you work in our wet lab, if you want to simulate using the computational thing, we can use this kind of a cellular computing as well, and that's what we have discussed with various terms. Now, let me go to the network biology. So, till now, we discussed something about system biology, that is, it is an integration of different types of omic subjects. What is this network biology? Network biology is, if a network biology is a part of system biology. In system biology, we have cellular computing, we have network biology, we have other areas of biology also in system biology. Okay, then what is network biology if it is a part of system biology? Network biology is a combination of biology, network theory, and statistics. Okay, the study of the combination of these three subjects, that is mathematics here, okay, network theory. Again, for network theory in mathematics, we study the form of graph theory. Again, this is a kind of a mathematics. So for network biology, one should be prominent in these types of mathematics, statistics, network theory, and biology, the general biology. The biology can be cell biology, molecular biology, immunology. All these are important to study about we integrate these types of biology in these subjects. Now let me take you to the main parts of network biology. Oh, this network biology origin, like earlier I told you, the genome. Okay, most of the important genome. What genomes? Human genome, uh, published in 2003, and rat genome in 2003, and mice genome 2002. So everything published in the 2005 itself. And after 2005, whatever we got the biological data in the form of a genome, the scientists start exploring on the proteomes. Okay, the proteome researches are still happening on, and you can able to see even no, not the genome project ended in this 2005. So whatever single genome is sequenced in this period 2003. Okay, now presently we are also having databases for 1,000 genomes, 10,000 genomes, and even 1 lakh genome project is also is in process. Okay, so there is so much kind of developments happening in the genome as well as in the proteome, interactome, lysosome, and epigenome, and whatever it is. Okay, so these are some of the contemporary biological applications. Now, something about networks. So, if you see the types of networks, generally we can observe protein protein interaction network, protein DNA interaction network, gene correlation network, gene regulatory network, metabolic network, and signaling networks. We see these examples in the subsequent slides. Before going to that, we see the types of networks. If you see this kind of a dots, okay, each and every dot, if it's a protein protein network, these are proteins. And if it's a gene network, gene gene interaction network, so these are genes. And whatever we call this dots, okay, the genes or proteins, okay, it can be genes or proteins. So according to the network, we use a term called node. Okay, this is what we call it as a node. The genes and proteins are classified as or categorized as nodes here. And whatever the line which is connecting the nodes is what we call it as edge. The nodes are generally genes or proteins, and the edges just connected. If you see the edges lengths vary with respect to each other. If the edges length are not constant, we call this as random network, and this is something like scale free network. Okay, and in hierarchical network you can see in hierarchical manner. In scale free network, so we have an equal length, but we don't find a kind of it is a you find a scale free network here. So that's what it distinguishes from the random network. And if you see this metabolic network, okay, the metabolites are connected to each other in this metabolic network. To see the metabolic protein interaction gene regulatory network, I suggest you to use a database called KEGG. KEGG stands for Kyoto Encyclopedia for Genes and Genomes. In this URL, we have a database called Pathway. So in KEGG Pathway, we can able to find metabolic network separately and protein interaction network separately. You know, whatever you are saying, this protein interaction network is yeast protein network. Yeast, we have 99% of genes, more than 99% of genes code for proteins. Okay, so you can see more proteins in the yeast in the small organism. But whereas if you see in human, okay, less than 2% of genes only code for proteins. Okay, in humans. But this example is for yeast. Okay, where even this organism we can find plenty of proteins and that are connected in this protein the networks. And this is a gene regulated network. In gene regulated network we can find two different colors, red color and black color. Okay, the red color here it implies the positive regulation. If a gene is positively regulating, we can find in one color, and if it is negatively regulating, we can find in another color. So it is most important to understand for a disease that just not the positive regulation is going to give a best target, even negative regulation. If negative regulation value, the p value of negative regulation is maximum, then even negative regulation gene is also a most important target. Okay, so one should also just understand, not just limit up to how the gene is interacting in the gene interaction network. One should also go to the gene regulatory network. That is something different from gene interaction. So only in the gene regulation we can distinguish the positive regulation as well as negative regulation, and even we can identify this through p values. Okay, each and every gene interaction with the hub gene. Here, the hub gene they mentioned is TP53, which is the most important target of cancer. So whatever interacting genes, interacting genes with the TP53, and this can also study through this regulation. So all these things we can study in system biology approach. Now something about network biology. In network biology, if you want to study about a network, just using two nodes, whether we can able to construct a graph? No, it's not possible. If you connect like this, we call this as a line. At least we should have a third node, node 1, node 2, node 3. If only we can able to have a kind of a graph, or we can call whatever graph we call, we can also call it as network. That's what you can able to see in this slide. But in this network, whatever you can see can be the node 1, node 2, node 3. See here, in this 2 to 3 connection, we have three different paths, A, B, C. Which is the shortest path out of all this A, B, C? Anyone can able to mention this is the shortest path. Okay, but we can also observe in gene interaction network, we can also observe the instead of following this B path, it can also we can get an A path or also like a C path. Okay, if so, it also depends here. Okay, it depends on how the gene is interacting with the another gene. Okay, that's what here they mentioned. You can't able to find a shortest path. You can also find the long paths also in gene interaction. And in this, if you see this third gene, okay, can also the gene itself can also interact with the same gene. For example, that some proteins like IL-4. Okay, the IL-4 ligand, if it interacts with the IL-4 receptor, okay, it can proliferate the T cells. So for T cells, IL-2 is based, and IL-4 is also based for dendritic cells and the antigen presenting cells. So the antigen presenting cells in terms, just because it's going to proliferate, even these cells is in turn is going to proliferate the T cells. So this is having a kind of a cyclic effect. That's what here you can understand. Self-looping can have a cycling effect in changing interaction. Obviously, the cell proliferation will be continuously increased. And this is something about the networks, the types of networks, biological networks. We already discussed not simply restricting up to the genome study can give a more understanding on system biology. One should integrate with the proteome, where we should know something about protein-protein interactions, and we should also integrate with the metabolism, that is metabolomes. Example for is protein-protein network. This is a kind of a gene regulation network of sea urchin. For sea urchin, we can see this red color, green color. If red color stands for positive regulation, green color stands for negative regulation. And this is metabolic flux analysis of E. coli. Metabolites can be found here, glucuronate, fructonate. So these metabolites you can able to see in this metabolic network. And you can see even the metabolic pathways can be integrated. For example, glycolysis can be linked with the ED pathway, and this can also be integrated with the pyrophosphate pathway. So whatever we see this, okay, the gluconeogenesis, glycolysis, citric acid cycle, this can be integrated with each other. So these kind of networks can also be available, and of course, these are important to understand about the metabolism. If you want to produce a kind of a metabolite from E. coli or any other organisms, one should understand this kind of how one pathway is going to integrate with the another pathway and how the metabolite is linked. You know, whatever product pyruvate we get from the glycolysis is like a input for the TCA cycle. The output of glycolysis is an input of the TCA cycle. Similarly, you can find for the other products also. So to understand about that, we should integrate the pathways. So studying about the pathways and integrating the pathways, everything comes under a part of system biology. So this is something about useful softwares. So this is a popular software, a freeware for visualizing any type of networks, protein-protein interaction networks, gene-gene interaction, gene-gene regulation. For all the types of networks, one can use this freeware called Cytoscape. Other than that, Pacheck and graph-related programming, one can use LADA, Naughty. And these are some of the network databases. I believe we have seen in the first session itself. What is string DB, what is Stitch, how these databases are developed, and what is the area of biology we are studying about this. You know, this is nothing but system biology. So this is nothing but system biology. So fun group, this is another pathway database, impact 2.0, network analysis, network analysis. These are examples of network databases. So now we can discuss something about network pharmacology. So we know that network biology and including a part of a network pharmacology is a part of system biology. So I told you generally this network biology or network pharmacology, this can be applied for treatment of any diseases. For bringing a precise medicine, we can use this kind of an approach. Even we have the polypharmacology. Okay, so first we study about the network pharmacology. So what is network pharmacology? Here, whatever we take a plant, okay, so from a plant we can have hundreds of compounds. From hundreds of active compounds, we try to screen it using any of the filters. The filters can be drug likeness filter, or it can also filter the active compounds based on ADME, including toxicity, okay, ADMET properties. We can also filter based on binding, like if you have a target receptor for a cancer, and out of this compounds, okay, which is going to bind effectively with the target receptor. So even this we can use it as a filter. So out of hundreds of compounds, we can filter to narrow down to one or three compounds, whatever best three compounds we get, and whatever compounds we get, okay, we identified as potential lead candidates. We take it for docking, binding analysis. And we can also take it for further pathway analysis, including disease analysis. Okay, and we can also construct the networks. So I have a few papers published in network pharmacology in reputed journals. We can also discuss this as case studies. So functional enrichments, I think we have discussed in the class one itself. String database, how to open the pathway database and how to select the filters and how to find the genes. Now, if I select the liver, okay, for a liver, so what are the important genes connecting to the HSP90? We can get these kind of a proteins. The red color proteins are connected with liver. Then how about this gray color? This gray color is not connected with liver. So like this in a pathways, we can select the filters and through the filters we can able to identify through colors. So we can able to see the important proteins found to link with HCC. From the network pharmacology, we found this as TP53. 23 percentage of the hepatocellular carcinoma patients are diagnosed with this important gene. So these are the other genes responsible for HCC. And the compound interactions, okay, can also be found from another database called Stitch, where vithafrene is a plant compound. It is interacting with these proteins, PAWR, EF2A, and similarly, you can find that drugs indomethacin, celecoxib can also interact with the protein called TP53. So only these drugs can also be used for treatment of cancer. Okay, so this is something about that, and in this is an example of cytoscape visualizer. So you can't able to use the output from the database. You can't able to put this in your journal paper, but whatever you have in your cytoscape visualizer, you have rights to publish in this journal paper. So I suggest you to download the networks and you just have your kind of visualizer interactions, and then you can take this for your publication. Okay. So this is a cytoscape visualizer opening the network here, and we can also have uh, thousands of plugins in this cytoscape to interact with the system biology approach. Okay, and if you see this database called Impact, this is an India's developed database for plant molecules. If you want to check about the drugs, okay, from the plant based source, this is the largest database developed by India, indigenous database called Impact. Okay, and that's the URL. And even in Impact, we can find this kind of a networks. Okay, like how a plant is associated with treatment of various diseases. So you can see one plant is associated with mouthwash. The same plant is used as a contraceptive, can be used as anti-infective. It is also used for treatment of fistula, rinderpest. So one plant can have thousands of compounds. So whatever we have thousands of compounds, okay, we have to isolate a specific active compound for treatment of particular disease. That active compound can be used for treatment of a few diseases, but not all the diseases. So one should identify the potential compound from a plant, okay, to treat all these kind of diseases. See, one plant is treating more than 100 types of diseases. The only thing is that you should identify the compounds. For this, even you can able to use this impact therapeutic use database. This is a herbarium. And if you see, this is a Chinese database called Herb 2.0. In this, we can see vithafrene A is present in center. Vithafrene A is a plant steroid from ashwagandha. So this is interacting with so many kind of proteins. Okay, so we can see this kind of information from this Chinese database, like how we find that information in India, and how about the gene interaction. All these things we can find here. Now we can discuss something about network analyst. Network analyst is another database. Of course, all these things are products of system biology. Okay, so in network analyst, we can able to study the multiple gene expressions. So we can study this through this online tool. This is a comprehensive gene expression profiling via network visual analytics. This is integrated with R programming. So one can able to use this express analyzed R. This is one of the tool which we can use it for differential gene expression analysis. For system biology approach nowadays, we won't simply uh, design and manufacture a drug for a particular target. We whatever drug we design, we try to check with multiple targets, and we check especially whether it is interacting with the important gene. If a patient is diagnosed with a gene, we are responsible for cancer. Whether this drug, whatever we develop, it is targeting that important gene. Also, it's targeting the interacting gene with the important genes. So all these things we study in this system biology. So for this, we can also understand the expression patterns. Like in the stage of cancer, there are we can find 4 to 5 stages of cancer. Whatever we find the stages of cancer, if you want to measure the expression, whether they are in second stage cancer or third stage cancer after treatment of plant compound. Okay, so one can also able to analyze this using this express analyst, or this is an integrated tool of network analyst. Okay, so whatever you see here is the R programming. So in R programming, they are introducing a library, they are using the functions, and they're executing it. So one should also have a R programming knowledge to work on producing this kind of a graphs. This is one such output. Whatever you see in the bottom is an output of R program. And there is another software called MetaboAnalyst. So in MetaboAnalyst, we can discuss on, this is the latest version, MetaboAnalyst 6.0. We can able to see the data of raw spectra to biomarkers, patterns, functions, and system biology. Okay. So for this, you can go to this URL, metaboanalyze.ca. So about these hyperlinks, I think I believe that I already provided in the first day session itself. I'm integrating this with system biology approach. And this metaboanalyzed CA. So this is from a Canada-based server where you can able to see this kind of integration of the data of MetaboAnalyst. And this is one of the most important DCHI network. Till now, whatever we discussed, this is something related to gene networks, protein networks, metabolite networks. But this is something related to disease gene networks, DCHI net. So this is a gene disease association network. For this, we have to go to teachinet.com. So here, if you see, sorry, that is repeated, diseasome. If you see this teaching network, we majorly study on diseasome, like how we have genome, proteome. Okay, here we study on human disease networks. That's what we call it as a diseasome. And the first published human disease network looked at genes, finding that many of the disease associated genes or non-essential genes, as these are the genes that do not completely disrupt the network. Okay, but which is passed to down generations. Many of the cancers are hereditary based. If so, what genes or carry forward which can cause cancer in future? So these genes can be identified, and this can also be edited based on CRISPR-Cas9 technologies, and the gene the disease can be avoided. So for this, we can use the diseasome. Okay, in diseasome, we have shared gene formalism, we have shared metabolic pathway formalism, and this is comorbidity, that is using phenotypic disease networks where two diseases are linked. If the observed comorbidity between, for example, if you see autoimmune diseases, the example, the best example for autoimmune diseases, arthritis, asthma, psoriasis. Do we have any connection between these diseases? This is something related to bone joints pain, isn't it? In bone joints, if you feel a pain, we call it as arthritis. We have types of arthritis. The popular is rheumatoid arthritis. And similarly, if you see asthma is a respiratory disease. We have the beginning stage to the chronic stage of asthma, the respiratory that is pertaining to the lungs. Okay, and similarly, psoriasis is something related to skin. But all these three diseases are linked with each other. Most of the gene targets, whatever we take in all these three diseases, are same. Out of 100 targets, one or two targets alone is different in arthritis when compared to asthma. Similarly, in psoriasis. So, like this, we can see the autoimmune diseases targets are almost same except a few genes. Okay, then you find a confusion. That's what we call as disease comorbidity. Okay, so this kind of a disease network can give a clear understanding about distinguishing the type of genes and even how the genes are connected, okay, in this type of diseases. So here also in the gene network, the networks can be visualized using the Cytoscape app, and this can also be R programming can also be used to understand various outputs, including generation of networks. Whatever networks we see in String database, Stitch database, this can be generated from the R. Okay, this can also be generated from MATLAB. Okay, the MATLAB, and we can also use the programming like Perl and Python programming. So we're just using the networks, but also constructing the networks, one should have these kind of a knowledge. And we can also associate with multiple diseases, where we can see pleiotropism, etc. Okay. Now, if you see about the diseases, okay, in this Tichy network itself, we have these two types of index. One is disease specificity index. What is disease specificity index? This is a value which we calculate in the Tichy network itself. These are genes that are associated with multiple diseases. For example, if you see the types of cancers, plenty of cancers are available, isn't it? But the most common important protein target is tumor necrosis factor. If any persons have more kind of TNF alpha, they are very less prone to get cancer. If TNF alpha concentration is more in blood, they are very less prone to succumb to cancer. And similarly, so this is something like a common kind, okay, common kind of a protein receptors, a family of protein receptors that play an important role on cancer. And especially if you see this disease pleiotropy index, here it is nothing but if the multiple diseases associated to the gene, multiple diseases associated to the gene or similar among them. Okay, so for example, just now I told you about the autoimmune diseases. For most of the autoimmune diseases, most of the targets are same. So in that case, we measure through disease pleiotropy index and disease evidence index. So all these things we can able to study in this Tichy network. Okay, in Tichy network we can find all this information. Cytoscape, I already mentioned you. Cytoscape is a open source bioinformatics software to visualize types of networks, especially molecular interaction networks. And we have plenty of plugins to work on various applications in system biology. Okay. For this, we need this kind of a language, extensible graph markup language and modeling language. And now let me take you to this uh, apps in Cytoscape. There are plenty of apps. If you go to the cytoscape.org, one can able to download and install the apps. If you already installed the Cytoscape, this kind of plugins will be embedded, and you can utilize the functionality in the Cytoscape visualizer. So apps in Cytoscape. So these are some of the graphical icons for each and every plugin. So we can also work with disease gene network. We can work on omics analyzer. We can work on the different types of gene interaction, protein, etc. So cytoscape, here we can see how we get visualizing the yeast gene interactions. This is an example of how to visualize the colorectal cancer. So this is a demo file provided in the cytoscape. If you open, you can find the visualization like this. So in system biology approach, the most important thing is that we have to take thousands of genes and we have to take screen the top 10 genes, something like that, as a most important target. So don't just restrict up to a single gene that this gene is alone is responsible for causing the cancer. You have to take thousands of genes and then we have to take consideration based on the expression patterns, gene expression patterns from the patients. You know, in GPA server, the data, we have plenty of data sets deposited from multiple hospitals in USA. In USA, so many kind of clinics are available, isn't it? Mayo Clinic, Anderson Clinic. So each and every day, multiple patients, I mean, many patients are approaching for the treatment. And so whatever drugs provided and based on the gene expression happen, all these data are available here. So one can able to find this information data set. Using the data set, one can able to find what are the most important genes in that particular, that is the patients are diagnosed with for the particular type of cancer. And we can also go with the gene enrichment using enricher server. So all these things are pertaining to system biology. So today I'll be circulating these journal papers to you, okay, to in the WhatsApp web. So this is pertaining to the system biology approach. So this is a paper published in 2020 in Informatics and Medicine and Law, a reputed journal. Here initially I tried taking top 100 differential expression genes, that is top 50 upregulated and top 50 downregulated genes from thousands of genes. So how I have taken this top 50 up and 50 down based on p-value, and based on the fold change. So based on these two properties, I have taken hundreds of differential expression genes out of 1,000 genes, and then I have gone with the top ranked genes and I enrich based on the pathways. If I'm focusing on some pathways, or like see these pathways, we have PA3K, AKT signaling pathway, RAS signaling, RAP1. So we have plenty of pathways responsible for that feminine cancer, ovarian cancer. So for this, whatever is most significant, okay, so for all this, I have taken what are the genes responsible for PA3K pathway, what are the genes responsible for RAS signaling. Like this, plenty of pathways I have taken and of course published in the paper itself. And we can also see what is the most important gene and how many genes are connected to that. So all this information also is shown in that paper. On top ranked genes, the overall top ranked enrichment pathways, and what are the immune systems related enriched pathways. This is something we have taken from the databases, and of course we have used softwares like Cytoscape, MATLAB for getting this kind of outputs. So especially we get Venn diagram to integrate the genes. Okay, so this is something about top ranked genes associated with immune system related signaling pathways. So before getting top 50, I have taken top 100 up and top 100 down. Then only I screened up to top 50 up regulated and top 50 down regulated. So finally, what I did, I have taken the top 10 important genes out of the 100, that is 50 upregulated and 50 downregulated. So top 10 genes I've taken, which is responsible for ovarian cancer, which is observed in most of the patients in US hospitals. So that 10 genes I've taken, and then I just talk with the popular plant compounds. Whatever plant compounds I have taken, quercetin. The quercetin is majorly available in berries. Also, you can find in tomatoes. So in our finding, you can able to find the tomatoes are important uh, for the ovarian cancer, the feminine cancer, and similarly resveratrol. Resveratrol is a compound observed in grapes. So the grapes can also can have this effect. But according to this study, we can see the quercetin is targeted for 6 sites. The quercetin has shown negative maximum binding energy out of 6 out of 10 proteins. It has docked with negative maximum binding energy. So quercetin is the best compound we have proved. I think if you have any doubts, you can ask me. One participant has, I think, unmuted. If you want to ask anything, I have just 10 minutes. If you have any doubts, you can ask me, or else I can continue. Good. Actually, there is a lot of information in system biology. It's like an ocean. Okay, so I think now I can go ahead because you didn't ask any questions at present. Okay, this is the case studies too. In the case study one, I have taken top 10 important genes for ovarian cancer, but before taking top 10, I have gone with so many system biology approaches. Okay, like gene enrichment, and gene enrichment based on different factors. Then finally, I have docked three important compounds with that, and then I identified the quercetin is the best compound for the ovarian cancer. And similarly, in the case study two, this is published in scientific reports. Here, the major study is, of course, system biology, no doubt in that. But here, I've taken stage one and stage two cancer. So, what are the most important genes responsible for causing the stage one and stage two kidney cancer? For these genes, I have gone for various system biology approaches. Okay, like using the pathways, using the top 100, top 50, whatever I work here, also we work. And we identified the most potential genes. Now we come across synthetic biology. What is synthetic biology? So, more about the system biology and synthetic biology softwares, even in the next session. Okay, so we'll be discussing. Okay, well, in the next session, that is on tomorrow, I'll be explaining you with the softwares. Okay, now let me first clear this kind of a theoretical information. Before, without clearing this theoretical information, if I take you to the software, you won't understand anything. So now let me take you to the next area of biology, that is synthetic biology. How synthetic biology distinguishes from system biology. Synthetic biology is an interdisciplinary branch of biology and engineering. See the difference there. There we have all the types of omics, but here you find engineering. Engineering, not just biotechnology, even the other areas of engineering. What are the other areas of engineering we use? We use electrical engineering, computer engineering, control engineering. So all this engineering, including genetic engineering, okay, comes here in this synthetic biology. So one should have a knowledge on these areas of biology and engineering. Okay, actually, what is synthetic biology? This applies the discipline to apply artificial biological systems for research, engineering, and medical applications. So to understand very clearly, I am going to give some examples. So, did you hear about the hybrid kind of guavas? Hybrid guava. Guava, it is a highly enriched protein fruit. If you see in all the fruits, guava is the in guava you can find high protein content. So before 10 to 15 years, we don't have that kind of a hybrid, large kind of guava. We can't able to hold a large guava nowadays with a hand, isn't it? But in 10 years before, we have very small guavas. We can hold the entire guava in a single palm. But now it's not possible. We have a large sizes. So what exactly you can find difference between the raw guava and the hybrid guava? So this is something general knowledge, isn't it? So can we interact? So if only if you can interact, then I can give you the importance of synthetic biology and can go further on synthetic biology. Can someone can unmute and answer about that? What is the difference between raw guava, because the here the synthetic biology is applied, raw guava and hybrid guava? You want me to show the pictures? What is the difference? Whatever you can interact. So I'm not asking in technical, I'm asking something general. See, the hybrid guavas are very large in size, isn't it? So what is the major difference we can able to see? Here you find the seeds are very less, but in small guava, plenty of seeds. And in hybrid guava, if you cut the hybrid guava, you can find even the light pink color. So look and feel that is very good in this hybrid guavas. And how about the taste? The taste is also fine, and this hybrid guavas are crispy. Okay, like eating snacks, we can eat the guava. But how about that raw guava? No, it will find many seeds inside, and if you have a strong teeth, You like eating raw govas. So plenty of genetic engineering happened in this hybrid govas. The first gene, what you can able to find here, what gene is silenced in the raw guava? The first is producing plenty of seeds, so reducing the number of seeds. Of course, we can't able to find even the seeds in this hybrid guavas. If you see purely hybrid guavas, I mean, that's not a pure, of course. Hybrid is hybrid, but if it is a fully hybrid, okay, then you don't find even a single seed. Okay, so but how to uh, go for gene silencing for not producing the seeds? For this, we need one gene, isn't it? What gene? So we take a particular gene in the raw guava and we silence the gene. Number two, for increasing the size of the pulp of guava. So we have to increase the gene expression, isn't it, for of a particular gene which can increase the size of a guava. So that gene is also incorporated. And third, to get a decent light pinkish color, also a gene is incorporated. And fourth, for getting a crispy kind of a taste, again one more gene is incorporated. So we have nearly four important genes in this hybrid guava in compared to the raw guava. So for this, which area of biology which knowledge you should have? Synthetic biology. So in plant biotechnology, in especially in the plant research institutes, they work on these kind of phenomena. So the synthetic biology can able to study through synthetic biology open language. Participants, I believe that I have taken 5 minutes late. Am I right? So I can take some 5 minutes extra about the synthetic biology. So until I complete this synthetic biology open language. So for this, one can go to this sbolstandard.org. Okay, this is the URL. We can able to get about the synthetic biology open language. What exactly you can find in synthetic biology open language? So in synthetic biology open language, for the genes, different parts of gene, the promoter is shown using this symbol, and complementary determining site is shown using this symbol. Similarly, for ribosome entry site, you know, like how you find these kind of genes, part of a genes, if you open it in any of the visualizers, especially the synthetic biology open language visualizers, you can able to find the parts of a gene in these symbols. So it is easier to identify. So 5 prime sticky restriction site is like this, and 3 prime looks like this, but 5 prime overhang is something like this. So like this, you can able to see all these symbols are connected to the genetic circuit. Like however you find an electric circuit where we find different kind of open gates. Okay, like similarly here also you find, like however you find in electrical engineering, here also you can able to find all these parts of a gene are connected, and that's what we call it as a genetic circuit. Synthetic biology, you have a biological part which is a sequence of a DNA that encodes for a biological function, for example as promoters or protein coding sequences. Okay, like this we can see, and see you can integrate multiple genes. So this is an example, you can see a composite part. Okay, similarly you can integrate one more gene here. So this is a genetic circuit. Whatever we see, this is a simpler genetic circuit. This is a circuit one, and whatever you see in the bottom is a circuit two. But circuits are generally very large, not like this. This is a part of a circuit. That's what I can able to tell. And synthetic biology open language, we have these kind of an icons. Just click this icon and drag and paste it. You get these icons in this software. Obviously, can develop the synthetic gene, whatever you want to connect. So multiple synthetic genes can be interconnected through this. So basic and composite parts. So to get a lack operon, to get a kind of different types of operon concepts, we can able to make it out using the synthetic biology open language softwares. And these are the parts I already told you. Now, if you want to know more about what are promoters, a promoter is a DNA sequence that tends to recruit transcriptional machinery and lead to transcription of the downstream DNA sequence. So, if you're not confident on the molecular biology, especially on the synthetic biology terms, whatever represented here. Okay, genetic engineering. So one can able to study this using the synthetic biology open language softwares. All this information are taken from the software itself. Okay, these terminologies and what for the genes part are used. There is a illustration of this process employed to construct engineered therapeutic microbes. Presently we came to know that we have gene modified therapeutic mosquitoes. What do you mean by that therapeutic mosquitoes? So if these mosquitoes can bite us, generally the mosquitoes bite us and it produces diseases, isn't it? But these mosquitoes, whatever engineered in the labs, okay, if they bite, they can also remove the disease. They can take care of, they take the disease. Okay, such a kind of a therapeutic mosquitoes are also available. How this is done? Through the synthetic biology. So synthetic biology is not used just for the plants, used in insects. Okay, used in yeah, used in different areas where the human can able to use it, including the editing of gene, editing of gene through CRISPR-Cas9 if you want to integrate it. For all these things, even the synthetic biology is needed. Okay, and in the next generation sequencing, whatever we sequence the genes and we identify, and if you want to edit the genes, okay, to overcome the diseases, of course, synthetic biology is needed. And here in this second part, you can able to see genetic circuit construction. We can just click that parts of the gene and we can integrate in the form of a circuit. We can also use in the form of a genetic logic gates like AND gate, OR gate, NOR gate, and through that we can able to control. Like for example, in Goa, okay, what genes we want, what genes we don't want, we can take it in consideration and we can able to produce it. Even if we want to produce hybrid Goa with more seeds, we can able to make it up. Okay, but whether you like it, nowadays the persons don't like the raw, isn't it? Whatever is good, we don't feel that is good. So that is happening just because of this synthetic biology. And this is how we integrate with the computational design using this synthetic biology open language softwares and testing platforms. We can also test it using the organoids, cut on chip, and we can measure the efficacy. All these things are a part of synthetic biology. We can integrate and we can develop numerous systems, including therapeutic molecules. This is one such example called LactoShield. This is an output of an iGEM team from SVCE. In 2016, they worked on this. What is the lifespan of a milk, whatever we procure? So in Tamil Nadu, we have Avin, and in Karnataka, we have Nandini, isn't it? What is the lifespan of a milk? What is a shell life? The lifespan we can't tell. We should use this is the appropriate term. Shell life of milk. Can someone answer for this? No time, isn't it? So I can answer 2 to 3 days. Just 2 days. If you keep it in fridge, you can keep it for 3 days. But according to Lactose Shield, we can increase the shell life up to 4 months also. How this is possible? I already heard something about ultra packed milk. Similar to that, the similar kind of a concept. So what we made is we add peptides to the milk and we try to make long-term preservation. Okay, so this we did through the synthetic biology approach. Okay, and of course we use this, and of course even the students have received the silver medal from Massachusetts Institute of Technology. They are conducting this kind of a competition, iGEM competition. Okay, so that's it. So tomorrow we'll be discussing on how to use some softwares, okay, for the system biology and anything is yes, taste is also different. You're right. Okay, good. Any doubts you can ask me at present. If no doubts, thank you very much for your patience and interaction. So tomorrow we see the continuation of the system and synthetic biology, of course. Thank you.